Many snake species can endure months, sometimes years, without a single meal. To survive this starvation, they depress their standard metabolic rates by over 70%, entering a state of profound energy conservation. Cut off from external resources, they rely entirely on endogenous stores, burning through lipids first and then heavily regulating protein breakdown. They manage this internal supply so efficiently that despite severe starvation, they continue to grow in length. Eventually, they secure a meal. When they do, the physical intake is massive. A snake can ingest prey equaling 25% or more of its entire body mass in a single sitting. Tracking metabolic rate over time reveals an immediate shock. Within 48 hours of feeding, the rate explodes, increasing 44-fold to power digestion. To accommodate this, internal architecture adapts instantly. Visceral organs like the small intestine, which atrophied during starvation, suddenly double in mass. To study this extreme postprandial response, researchers turned to a specific model organism, the Burmese python. Within 24 hours of feeding, the python executes rapid, reversible organogenesis, generating new tissue in a fully mature adult to support the meal's metabolic demand. By extracting tissue and mapping RNA sequencing data across a 10-day feeding cycle, researchers have isolated exactly how this happens. The massive structural remodeling of the intestine is dictated entirely by sequential, highly orchestrated waves of gene expression. At time 0, 30 days after its last meal, the python's intestinal lining is completely dormant and physically atrophied. 6 hours after swallowing a meal, the prey remains in the stomach. If you examine the small intestine at this exact moment, there are absolutely zero macroscopic physical changes visible. These expression cluster graphs reveal a hidden biological explosion. Within this initial 6-hour window, over 2,400 genes are significantly differentially expressed compared to the fasted state. The data points directly to a specific molecular driver coordinating this initial burst, the WNT signaling pathway. RNA transcripts show sharp upregulation of the WNT5A gene and its extracellular receptor, frizzled. Simultaneously, intracellular inhibitors like axin 2 and TCF7L2 drop off the sequencing charts, effectively releasing the biological brakes on cell growth. This precise transcriptomic push and pull creates immediate promitotic pressure. The tissue is perfectly primed for massive cell division hours before a single cell has actually physically split. Moving into the 12 to 24-hour window, the genetic instructions begin to manifest as physical architecture. This cross-section models the first physical manifestation of those instructions, hypertrophy. The existing enterocyte cells lining the gut swell, expanding their individual volume by 2.6 times. Then comes hyperplasia. Massive cell division accelerates across the entire mucosal barrier, drastically multiplying the total cell count. The RNA data from this period confirms the mobilization of specific kinesin proteins, which are required to manage intracellular trafficking during these rapid anaphase and cytokinesis transitions. The ultimate consequence of this cellular restructuring occurs at the surface. Existing pools of cytoskeletal proteins migrate to the tips of the intestinal microvilli, causing them to elongate to 4 times their original height. This extreme lengthening vastly expands the internal surface area, creating the exact physical space required to process the massive influx of prey that is now exiting the stomach. In a single day, a hidden transcriptomic burst has successfully built a highly specialized, fully functional digestive infrastructure from scratch. By day 4, roughly 70% of the rat meal has been converted to chyme, exiting the stomach and completely filling the newly expanded small intestine. This chart tracks nutrient uptake over time, illustrating the direct result of that expanded surface area. Absorption rates for D-glucose and amino acids like proline and leucine curve sharply upward right at the 24-hour mark. Overall nutrient uptake capacity increases up to 20-fold compared to the fasted baseline. This physical absorption capability perfectly mirrors the genetic data. Expression charts from this exact window show the massive upregulation of hydrolase and nutrient transporter genes. This 4-day plateau represents the absolute peak functional state of the python's digestive cycle. Anatomical form is strictly dictated by physiological function. The genetic blueprint specifically timed the completion of the new tissue infrastructure for the exact moment maximum absorption was required. Past the 4-day mark, digestion concludes. The remaining chyme empties out of the small intestine. This creates an energetic trap. Maintaining this massive, hyperactive digestive infrastructure when there is no food left to process is a deadly liability. To conserve energy, the transcriptomic program abruptly reverses to dismantle the organ. This heat map matrix of gene expression reveals an aggressive transcriptomic shift at the 96-hour mark, the massive upregulation of initiator and executioner caspases, specifically caspase 3 and caspase 6. These caspases trigger highly controlled apoptosis, executing programmed cell death across the newly generated mucosal tissue. 10 days after the initial meal, the organ mass, the physical structure, and the entire gene expression profile have returned to the flatlined baseline. The python's visceral shrinkage is not a passive deflation caused by an empty stomach. It is a highly targeted, actively programmed biological self-destruction. This looping timeline illustrates the Burmese python's postprandial response, a masterful display of extreme metabolic economics. Its radical phenotypic plasticity is entirely governed by distinct sequential waves of gene expression. But these underlying genetic pathways, the WNT signaling network that builds the tissue, and the caspase executioners that tear it down, are not unique to snakes. They are highly conserved across all vertebrates. Humans share this exact cellular machinery for intestinal regulation. When WNT signaling becomes dysregulated, it can lead to uncontrolled intestinal cell proliferation. This is a primary driver of human pathologies, particularly colorectal cancer. These exact mechanisms are also deeply implicated in inflammatory bowel diseases like Crohn's and ulcerative colitis. The python's hyper-regulated physiology provides a comparative model for adult organ regeneration, offering researchers an opportunity to observe these pathways in a discretely staged, predictable cycle. Mapping these growth and regression triggers provides a framework for investigating how these same pathways might be modulated to treat intestinal disease in humans.